we have to give you something to thank you, and you can remember us. So this is from the Translational Molecular Imaging Institute, 5th Annual TMII Symposium, presented to Scott L. Friedman, July 24, 2016.
Wow, that's great. Thank you.
Thank you. So this is now the opening of the program officially with our keynote speaker, Dr. Roderic Pettigrew, MD, PhD, Director of the National Institute of Biomedical Imaging and Bioengineering at the National Institute of Health. You're good. So let me introduce very briefly Rod, who probably a lot of you know already who he is. So he is the first director of NIBIB, and officially really, I mean, almost now 2 decades, 2002, end of 2002, 2003. And really, he has been the first director to that institute. And that institute, started with a lot of doubts, from in the community, I think, but also a lot of support from people who felt that engineering and biomedical engineering and biomedical imaging needs to have a home. And Rod has really elevated the institute to an incredible level with incredible number of innovations that are coming in and translational work that we will hear about from Rod. Rod also in 2003 has taken a key role at the NIH as the acting chief officer for scientific workforce diversity, which is a very important topic here also at Mount Sinai for us and other places. Prior to coming to the NIH, I actually had the pleasure to meet and to serve under Dr. Pettigrew. He has been actually one of my first mentors, and I will talk at the end a little bit about our personal journey together. So he has been working in the field of cardiovascular imaging as professor of radiology of medicine at Emory and bioengineering at Georgia Tech. He's done incredible work in multimodality cardiovascular imaging with a lot of emphasis on 4D imaging using magnetic resonance imaging. And now his current research is trying to integrate the aspect of imaging and other aspect of biomechanical modeling for coronary atherosclerosis. He's also worked with other institutions such as the Howard Hughes Institute and NIBIB to promote the interdisciplinary graduate program, recognizing the importance of setting up such a thing. He also has done set up incredible program at NIBIB related to how do you push and how do you actually fund high-risk, high-yield research. The Quantum Project, I was actually involved in a few of these review panels that set up these really incredible medical moonshots to pursue these big projects. Also here at NIBIB and Rod has also helped a lot to set up the BRAIN Initiative and invest with NIH in the BRAIN Initiative. And finally, he also has worked internationally with a collaboration recently between the US and India to develop technology, point of care, simple technology that can be translated very easily worldwide to solve the problems of cardiovascular disease, like hypertension, for example. Rod also has been elected as member of the 2 most prestigious academies, the US National Academies, the Institute of Medicine, and the National Academy of Engineering, a well-deserved award. Many other awards have also been bestowed on him, which will take me actually a long time to quote. But also I wanted to say that he has been, for me, has been really early on, before he was the head of NIBIB, our path crossed because of our interest in cardiovascular imaging. And he has been extremely generous with me, helping me, giving me advice, how to get actually my first R01. And I remember, Rod giving me a call, and also when he leaves a message for you, Rod, it's very dramatic. You feel like there's something. So I would always be scared, what's going on here? Would you tell me, Z, I need to talk to you? I was like, oh, okay, let's make sure we talk to Rod. I've also served, and he mentored me early on, also serving the ISMRM in several committees within ISMRM, SCMR, AHA, etc. So I've always taken his advice. His advice has always been right on. And also, I early on, having interaction with him, I always wondered, why would he do something like this? And it really is testament of really his care, to mentor and train people. So I appreciate that, and it's so great to have you here, Rod. So without further ado, we will be looking forward to hearing from you related your presentation.
Thank you, Zahi, and good morning, everybody, as I get set up here. Great pleasure to be here. This has been a long time coming. Zahi, Z as I call him, that's accurate. It did not embarrass me by telling you how many times we've talked about my participating in a symposium over the years. And it isn't until now, I guess the fifth time, that I was finally able to make it. Really delighted to be here. Had planned to come in and take the next flight out right after my talk until I saw the lineup on this program. And I really have to compliment Zahi for the quality of the program and the stellar lineup of speakers that he has. So I changed my flight, actually. I have a travel coordinator who had made that arrangement, and when I saw the program, I told her to change it. I'll be here for the whole day because of the lineup of the speakers and the opportunity that I have to learn, which I really enjoy doing. So that introduction is a tough one to live up to, so I think I'll just forget about it and step right into the presentation. I have modified the advertised title a bit without knowing what Z would say and also what Scott would say. And my modified title is, as you can see, Imaging Innovations and Precision Medicine. So there are two, I think, key terms here, and those are innovation and precision, particularly focusing on precision measurement in order to facilitate precision medicine, or the themes that I will explore, and delighted to do so from my perspective as an NIH Institute director, and perhaps less so from an active investigator. With the lineup that you have, I think my home base, which is cardiovascular MR, is well covered by one of the speakers on the program, and also one of the audience members with Matthias Stube here, who's speaking, and Dan Sodikson, one of your neighbors down the street, who are both experts in this area. You will certainly learn from Matthias and perhaps from some commentary from Dan, and with the other stellar lineup that we have on the program. So without further ado, I want to begin by talking about precision measurement and how important this is and will be in delivering on the promise of precision medicine. And this concept of precision measurement and how important it is to discovery, how important it is to innovation, dates back to the beginning of the scientific revolution with Galileo. And we all know, perhaps, the storied history that Galileo has. He was one of the people that really put forward the concept of the sun being the center of the universe rather than the earth, as was believed at that time. And you may recall from your high school history that he was in fact imprisoned, placed under house arrest by the Catholic Church because he spoke heresy, which was to say that the earth was not the center of the universe, that rather the earth moved around the sun. But perhaps more to the point of his contribution to showing how precision in our observation, precision in our measurement leads to new discoveries is something now that we all well know and take for granted, but in the 1600s, this was quite a revelation. When at that time he debunked a commonly held belief, which was that the Milky Way was a nebulous cloud, like the cloud that you see in this image before you, but used the telescope, which was a new instrument at that time, had recently been invented, to make more precise observations and measurements. And on the basis of that, of this early stage scientific revolution imaging, observed this in his famous quote here: "The Milky Way is nothing else but a mass of innumerable stars planted together in clusters." And this was a shocking but fundamental revelation in the 1600s. We now move forward to one of our current standout scientists, Stephen Quake, who is a professor of bioengineering at Stanford, won the Lemelson MIT Prize a couple of years ago for his work. Has this quote here that I'm particularly fond of. He also talks about Galileo and how he ushered in this era of precision measurement, and makes the statement here that by measuring fundamental physical phenomena with increasing precision, one can make amazing discoveries. And that's a powerful thing, that by measuring fundamental phenomena with increasing precision, you can make amazing discoveries, and even sometimes stumble, and I would say uncover rather than stumble, new laws of nature. If we move to the current day, another one of our leaders, that is a president, has similarly now discovered this move towards more precision in our observations, more precision in our understanding of disease, and therefore more precision in our ability to treat disease. The concept of precision medicine, which has been discussed for the last several years, and now championed by none other.
So tonight I'm launching a new
than
precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. We can do this.
And as you can see there, it's broadly supported by all of Congress. So I like this simple, plain speak, layman's description of what is meant by precision medicine in the broader context, delivering the right treatment at the right time, every time, to the right person is what we are after. And if you look then at the challenges that we have in achieving this goal, the right treatment, right time, right person, every time, there are a number of challenges across the broad medical spectrum which will require technological innovation in order to meet these. And some of those, the key ones, are listed here. We know we have rising healthcare costs, so we need to restrain costs while increasing quality. Chronic disease is on the increase, consequent to our success in prolonging life. We now add about a year to life expectancy every 5 years, or 6 years at birth. We certainly now live in a global society. The world is super hyper-connected. We don't think about regional issues anymore the way we did just maybe even a couple of decades ago. We all think about what's going on around the world because the world are we. And this is another challenge, to be able to address diseases that are global in nature. And with our increased knowledge of molecular science, then we have this opportunity to deliver on this promise of precision medicine. But the challenge is to do all of those things, to be more precise, broader, meet this growth in chronic disease, and to do that in a more value-based paradigm. These are competing demands, I should say, and meeting those competing demands requires that we leverage all of the sciences, the life sciences, the physical sciences, and engineering. And I was so delighted to hear Dr. Friedman talk today about this medical training program and the innovation that you have here, where you're folding in engineering at an early stage into the medical training paradigm. Our solutions to complex problems will come from attacking them in a way that they are constructed. And complex problems don't have within them these neatly defined scientific disciplines that draw borders between physics and engineering and math and chemistry. In nature, they're all together, they're seamless. So our solutions to these has to be based on an approach that integrates all of these sciences that we study. This point and the issue of bringing engineering more into medicine in order to achieve this goal of translation is a practical one, and was the point of this opinion piece authored by several of my colleagues and myself recently in Science and Translational Medicine. You can see there, Sue Chin, Rashid Bashir, Bob Niram, and myself, where we made just this point. And one of the concrete examples that illustrates how important engineering now is to translational medicine, I think, is shown by this graphic, which comes from the NIH data. And what is being shown here over the last 15 years or so, reference to the year 2000, is a percentage change in money, and three kinds of money. The first is the funding of the NIH, and you can see that's in gold here. So referenced to 2000, the NIH funding has been fairly flat numerically over the last decade. If you account for inflation, it's been a functional decrease, but numerically you can see the flat amount of funding over the last decade. Contrast that, however, to NIH funds that have been awarded to two different areas, engineering departments as a whole in blue, which has increased fourfold, and to biomedical engineering departments, which has increased fivefold. Rather dramatic, and actually when I saw this, even I was surprised. But I think it underscores the importance of engineering in achieving our broad medical goals, and therefore the quality of the application of the science that come in that include these, and as a result, the increase in funding to bring this more into the whole discovery and translational process. So if we talk then about the value of imaging innovations in achieving this mission that the President now has taken on of delivering on the promise of precision medicine, I'd like to illustrate this in three categories. Imaging to uncover new fundamental knowledge about disease and disease mechanisms and biology, turning that discovery into health, and then continuing to push the frontiers of discovery through advances in imaging precision, looking first at making new discoveries through more precise imaging. And rather quickly, this was, to my knowledge, the first time that I am aware of where a gene that places you at risk for Alzheimer's disease was discovered by imaging, and in this case, imaging performed necessarily with great precision in order to uncover an association between a particular gene and the risk and presence of Alzheimer's. So the specific gene, the genetic variant that was uncovered was the SPON1 gene in this particular variant. This was done by way of high angular diffusion tensor imaging, looking specifically at fractional anisotropy in patients who were a part of the broad Alzheimer's disease neuroimaging cohort, some 360 or so of them, and correlating the density of neural fibers between regions of brain function. And those functional regions are color-coded in the top, the fiber density by fractional anisotropy shown here, and then the extent to which increased or decreased fiber density correlates and then correlates with genetic expression is shown here. And by way of this plot that you see here that indicates all of the genes, and on the vertical axis, the coefficient of a probability coefficient P that indicates whether or not, and there is a correlation with a specific gene and the density of neural fibers between any given region as shown in this particular map. And what is seen right here on chromosome number 11 is this large peak. In fact, those of you who are familiar with these kind of plots know this is somewhat ironic talking about this here, that this is referred to as a Manhattan plot. And it's a Manhattan plot because it does look like the Manhattan skyline. Well, right here at chromosome number 11 is this p-value that is 10 to the minus 9 in terms of the significance of a correlation between fiber density and this specific gene and gene expression, which is a variant of SPON1 gene. If one has this variant of the SPON1 gene, it is actually a gene, that variant is protective. So having the gene places you at risk for Alzheimer's. Having a mutation in that gene, and those individuals that are fortunate actually to have a mutation in that gene, correlated with a much lower incidence of cognitive decline and Alzheimer's symptomatology. So this observation having been uncovered by precision imaging, as I would place it. There may well be others in this audience that are even more familiar with than am I, given the, as you pointed out, that I think, Z, that about 50% of the faculty focus on neuroimaging here. So here's another finding when I first saw this I thought was fairly striking, but also points to precision by way of quantitative measurement and making new discoveries about disease. This is a map shown here in color, where the color corresponds again to the p-value in schizophrenic patients in whom a specific biomarker was, an imaging-based biomarker was assessed for those individuals who were schizophrenic in comparison to community controls and also in comparison to relatives who did not have psychotic illnesses or episodes. And the specific marker is tensor-based morphometry, in which this tensor-based analysis looks at volume and volumetric changes over time following these schizophrenic individuals over a period of several years. And what you will see in the movie is the change in the progressive decline in regional volume, how this occurs and occurs in a wave, and the extent to which that is significant in comparison to the controls. So in plain speak, the pinker the area is, the greater the regional volumetric loss. And again, you can see how this progresses over time, indicating that individuals with schizophrenia actually lose regional volume, particularly in the frontal temporal and frontal parietal areas, this sort of frontal temporal decline in volume, which more recently has been shown to be ameliorated by some of the antipsychotic drugs. I think stunning findings to me, and in particular when I first saw this. But again, because of the precision, uncovering this new information, and with this new information, then armed with our ability to evaluate interventions. Here is another remarkable example that I'd like to show you. Some of you may seen this before. I'm particularly fond of it. Let me set up this video that I'm going to present. Here's a lady in the operating room with severe depression. She's being treated with deep brain stimulation. And the electrodes that are being used to stimulate her are placed in an internal capsule. At the top, superimposed over her face in white numbers, will be the voltage of the DBS. And as you listen to her, you'll hear her rate her level of depression as the voltage is being modulated.
And let me know if you feel anything different, Karen, okay? How you doing, Karen?
Okay.
You notice anything different at all?
No, not really. I feel okay. I feel pretty good.
Better than you did before? Or the same?
A little better.
What are you feeling?
Happy.
Happy?
Yeah, I feel good. I just feel really good now.
What?
I like to smile.
You want to smile?
Yeah.
Do you
I haven't
notice?
smiled for a long time. Yeah.
Smiling again. Laughing. What do you feel like now?
I feel pretty good.
Where would your mood be now? 7 was before.
Yeah, well, maybe
About the
an
same?
8.
Okay.
I feel pretty good.
Kind of laughing, huh?
What are you thinking of right now? What are you laughing for?
Yeah, that. Because I feel so good right now.
How you feel now, Karen?
I don't feel like before.
Where's your mood at right now?
I would say it's back to 5.
What are you feeling, Karen?
Okay, so a rather obvious, strong, direct, and immediate correlation between the strength of the stimulation, the level of voltage, and her mood in a person who said that she hadn't smiled in years, in a long time. And now suddenly she's smiling when she's stimulated with a high level of voltage. When I first saw this and discussed it with the neurosurgeon Gerald Vitek, I go, this is amazing, you should use it more. He goes, well, it doesn't work all the time, and we can't figure out why. You know, when it does, it's like this. It's no question about cause and effect, but we can't get it to work all the time. Well, we may have an answer now, and the person who pioneered this approach for intractable depression is Helen Mayberg, and she writes here, neuroimaging-based biomarkers for treatment selection in major depressive disorders. And the discovery is, again, somewhat stunning initially and somewhat surprising, but perhaps not really. What is shown here is now our ability to identify neural fibers in tremendous detail. This comes from the work of Van Wedeen, who demonstrates not only the identification of the fibers, but the crossing structure of the fibers in this grid-like pattern. which resulted in the cover of National Geographic just last year. And with this increased ability to resolve these fiber crossings, again, with high-density spectral diffusion tensor imaging, as he calls it, looking at diffusion in 300 isotropic directions is what he does, identifying now the intersection of 3 fiber bundles, and with this latest study, determining where the sweet spot is for stimulation to actually achieve a successful therapeutic approach using deep brain stimulation. So she writes here, defining critical pathways mediating successful DBS for treating treatment-resistant depression. That is, identifying these fibers and where the 3 of them intersect, which is variable from person to person. She makes that point, saying here that gray matter is variability is not the source of variance. Rather, it is the variability in the hub location where these fiber bundles intersect. And with this kind of precision imaging, being able to identify where that is. So the successful approach, I think, is based on this, and now we perhaps understand that. So that kind of new information. Looking now to translating this even more so into frontline treatment and management of various diseases, a couple of examples here. So here is a simple question in the OR. In an individual with head and neck cancer or prostate cancer or breast cancer, where is the cancer? What is the extent of it? Is it all gone? And in the case of head and neck cancer and prostate cancer, which is innervated by a plexus of dense nerves, where are the nerves in distinguishing those? Here is a video, another one, from one of our supported researchers that talks about an approach to addressing this problem.
Wanting to improve surgical outcomes for cancer, Quinn Wynn, a head and neck surgeon at the University of California, San Diego, teamed up with Nobel laureate in chemistry Roger Chen, also at UCSD, to develop a fluorescent molecule that causes cancer cells to glow during surgery, making it easier for them to be removed. The molecule has a horseshoe-like structure. One arm has a positive charge that causes it to stick to all cells in the body. The other arm has a strong negative charge that neutralizes the molecule, preventing the positive arm from sticking to a cell until the right time. Attached to the ends of the arms are 2 different fluorescent dyes that cancel each other out when in close proximity. When the molecule comes in contact with enzymes that are found specifically in tumors, the back of the molecule is cut, freeing the positive arm with its dye attached to it to enter the tumor cells. Here's how it would work. Prior to surgery, a solution of the horseshoe molecules can be injected into the blood. And once the surgery has begun, cells that have taken up the molecule can be visualized. Depending on the type of fluorescent dye used, different equipment is needed to see it. Clinical trials are expected to begin in 2015. But tumors aren't the only tissue worth illuminating. Wynn is also developing a near-infrared probe that labels nerve tissue, which can be difficult to identify during surgery.
And then speaking more specifically about distinguishing tumor from nerve, not only making the tumor and tumor margins more visible, but distinguishing those from nerve. Here we see this tumor in a breast cancer model, which is made to fluoresce, and you can see the tumor tissue here. Here's a nerve entering it. Then labeling separately and with a different fluorescent probe the nerve, you can see much more clearly the distinction between the nerve, how it invades the tumor, the relationship between the two, and therefore facilitate a surgical resection without damaging the nerve. Critical, as we certainly understand in prostate surgery, but also in head and neck surgery. Here's an example that really is an exemplar of the importance of the merger of engineering, the physical, and the life sciences, and moving closer towards delivering on the promise of precision medicine. A better way to identify and monitor cancer, and therefore deliver on cancer treatment, would be facilitated by having the ability to, in real time, identify individual cancer cells as they are leaked from the tumor and shed into the blood. Now, when this project was undertaken, the thinking at that time was that this would occur, that is, cancer cells in the bloodstream, in rather advanced tumors, but perhaps before they metastasize. So the intent was to prevent metastasis. But what has been learned through this research is that circulating tumor cells may be present even in early stages before the cancer has left its local and primary site. This seems to be the case in prostate cancers being explored. But here is a innovation which is a handheld, point-of-care, microfluidics-based device that allows one in a routine blood sample to identify rare, early-stage circulating tumor cells, these CTC cells, at a sensitivity of 1 tumor cell per billion normal cells. When done in a given person, this provides the potential to make a individualized prognosis, determine the most effective therapy, and then, of course, logically, the monitoring response. Here's the platform technology that you see here, diagrammatically with multiple stages where a whole blood sample is placed in these separating stages for the blood, whereby inertial forces and filtering these small components, red blood cells and platelets, are filtered out. Then the white cells are tagged with magnetic beads. We know a lot about white cells and how to target their surface proteins. So in this magnetic field gradient station, pulling the white cells over to one side and then leaving the circulating tumor cells in the residual port. An inexpensive chip, extremely sensitive, 1 cancer cell per billion. Here is a video of this in process, with the white cells being siphoned off to one side, the tumor cells being shown here. So the ability to detect these in a routine blood sample is big news in terms of early detection. But that's only the beginning of the story. The next part of the story comes in what's shown in this title. Now that you have those cells, you can study the biology on a person-by-person basis and test for drug susceptibility by way of genomic profiling and identifying druggable targets based on those profiles. So here is a display that shows a circulating tumor cell that has been cultured. And with those cultures, then the testing with this heat map display across a large range of potential drugs. In this one of multiple examples of different cell lines, and this particular cell line shown here is BRX07, characterized by genetic expression factors that are shown here. And these specific genetic genes that are expressed that characterize this particular cell line are then tested and give an indication as to what drugs might be used to target the genes responsible for the cancer expression. And what you see on the vertical column are these different categories of drugs that attack these specific genes, the PI3 kinase, for an example, and fibroblastic growth factor receptor is another example here. And in this particular display, the so-called heat map, each one of these tiles represents a replicant of the original cell line, post-culture, being tested for this range of drugs with a range of dose levels shown in these black bars, and being tested in quadruplicate, as you can see each of the four. The color shows the level of cell killing. So dark brown is no cells are dead, and in dark blue, all of the cells, 100% of the cells have been killed. So now it's quite clear which category of drugs and which specific drugs within that category for this specific cell line are effective against this particular cancer cell. And you can see here the most effective one is a FGFR category class of drugs, and each of these specific drugs being effective in killing the cell line at a relatively low dose. Very powerful because you're able to do this on a cell line by cell line basis, and we know that in individuals there may be multiple cell lines that are responsible for a given cancer. The most recent news, just published a couple of months ago, is that with a platform like this, but specifically engineered to do what is indicated here, and that is capture circulating tumor cell clusters, has considerable promise in our battle against cancer, because these clusters, when they occur, and they occur as a rare number of CTC cells, but at a fairly moderate prevalence in terms of number of cancer patients that have them. But when they occur, this is really bad news. Clusters have 20 to 50 times greater metastatic potential. They're associated with dramatically reduced survival. So when they are found, we have to be more aggressive in our monitoring and in our therapy. This is a different type of circulating tumor cell chip, specifically engineered to capture these clusters. They are more difficult to capture because they are fragile. You can't use the high flow rates. In the previous version, there was cell sorting. In this version, it's actually trapping, as you can see in this triad of triangles here, the cluster being trapped. But once identified, then the work really begins, and we really have to roll up our sleeves. The point is being made here in patients with prostatic cancers, in whom there were either single circulating tumor cells identified or clusters identified, and the differential survival curves for single cell individuals and CTC clusters in red. If those individuals only had single circulating tumor cells, you can see their survival was out to, this is about 4.5 years. This is shown in days. 1,600 days is about 4.5 years. Whereas those who were found to have CTC clusters, their survival was even less than a year. So dramatically important, an important finding. Just very quickly, also in the theme of turning discovery into therapy, this is on the horizon, and if it works out in humans, this will be a great thing. This was shown in mice, where focused ultrasound was used in a transgenic mouse model of Alzheimer's disease and found to have considerable therapeutic promise. What is shown here, I'll just summarize by saying, in those mice treated this way with focused ultrasound, and the observation that was made was increased neuronal development. Newborn neurons were developed to a much greater degree in these mice that were so treated. And that's what's shown here in the black bars. These are in the controls and then also in the mouse model of Alzheimer's disease. This is the number of new neurons found in those post-intervention in the black bars. Achieving this neuronal stimulation both in the controls and even more dramatically in those mice with Alzheimer's disease, as well as the length of the neurons. So I think a very exciting finding, and if that translates into patients, this would be one of the first types of therapies that we have to tackle this very crippling disease. So I'd like to conclude with a couple of examples about pushing the frontiers and innovations in the precision measurement arena, precision observation arena, that will allow us to make even greater discoveries in the future. The first one is in the area of super-resolution microscopy. This was the work that led to the Nobel Prize just last year in chemistry. Here are the prize winners. Two of them have ties to our institute. Bill Mourner was actually on our board of scientific counselors, and Eric Betzik, who early on was at the NIH, collaborated with two NIH investigators now, George Patterson, who is the second author on the science paper that actually resulted in Eric winning or sharing the Nobel Prize. And then Harry Schroff, who is one of our superstar investigators, who was the postdoctoral fellow of Eric Betzik at Janelia Farms. And both of them, both of those individuals, as well as Eric, have continued to work in this field and are pushing the frontiers of super-resolution microscopy, imaging live organisms at the nanoscale level. Here is a microscope invented by Harry Schroff that does selective plane imaging microscopy, avoiding phototoxicity with laser, which has been the problem in the past, is that when you stimulate a live organism with laser light sufficiently enough to be able to image them, it kills them. And so what he has done is to image individual planes, scan through the planes, do those in two orthogonal directions to get bidirectional views, sweep through the planes at a fast rate, 200 planes in about 1 second. And with that, he's able to now image living small organisms such as this C. elegans here and observe the embryogenesis of a C. elegans embryo. Now, for those of you who have not worked with this creature, it is tiny, tiny, tiny. You can barely see it with your naked eye. It's about the size of the end of a sharpened pencil. And here is the embryo in the egg sac at a two-cell stage, shown here, and then progressing over time over the 15-hour gestation period. You can see the progressive cell division. It begins to look like a worm here, the serpentine appearance. And now it breaks out of the eggshell, yolk sac, and here is the grown worm. Here is actually a video, I think, that is even more amazing in that within the embryo, imaging an individual neuron in a developing embryo. Here is a lapse time shown here. This is at 4 hours. So here's a neuron. You see it begins to sprout two pods on either side. They extend out. Now we're at 5 hours. The synapses here begins to look like a lobster, like this Maine lobster with these two claws growing out, now out at about 6 or 7 hours, whereas where this video stops. And beyond that, these neurons, if you follow them, they begin to migrate. So this is very repeatable. He's studied a number of these organisms, and the time scale by which this occurs and the place in which they occur is repeatable. His goal is to develop this four-dimensional map of the neuronal connectome of the C. elegans, as demonstrated here, but the question is, how does this occur and how does it happen over time? To establish that, and then once you establish that, you can start looking at what happens when you prevent it in a certain area and the benefit of interventions and how those would facilitate recovery of health when disease occurs. So we look forward to learning more. And the final thing I'd like to show you here is another remarkable innovation that has tremendous promise in this field. For those of us who have looked at coronary angiograms as the gold standard for imaging coronaries, we know that those are done at a frame rate of about 60 images or 60 frames per second. Lihan Wang has developed this approach that now allows you to image at this incredible frame rate, 100 billion, with a B, frames per second. I'll repeat that again. It just staggers the imagination. 100 billion frames per second. This is in the picosecond range, in the tens of picosecond temporal resolution, using a combination of a couple of approaches. Street cameras, which have been around before but do one-dimensional images, combined with compressed sensing, which the MR community is well aware of, to generate now these, for the first time, two-dimensional images at this incredible frame rate. And just a couple of videos to show you the kinds of things that you can now observe with this framing rate. This is a packet of laser light as it strikes a mirror and bounces us out. Just incredible. I've never seen this sort of thing before. You see the reflection of a packet of laser light. Here, so I'll stop this one. And this one, you can actually watch the refraction as it goes from air to resin. And to do so even quantitatively. Stop this one. And here is these laser packets racing through air versus resin. Again, being able to quantify that in this tens of picosecond range. You can see they start the same, and then the laser packet in air gets ahead of that in resin. And then perhaps my favorite one is this one, where a higher energy packet of laser light in the green spectrum strikes this fluorophore, and then you have downstream in terms of energy fluorescence in the red frame. So you watch this, it's going to happen pretty instantaneously. Boom, hits it, and it immediately fluoresces. So I'm going to stop this and try to go just to the point where that occurs. Boom, right there. So you see that? Of course you can see it. This green light strikes the fluorophore, and then immediately, boom, it turns into red. So, I think a tremendous development for the scientific community. The potential applications include uncovering things like being able to explain and observe neural conduction, synapse function, cell activation, being able to decipher how neural networks work in health and in disease, or immediately come to mind with some other things, right, as you can see here. The intermediate stages of molecular targets for drugs. Cell proteins fold and unfold, and we target them with maybe 50% of the drugs, G protein coupled receptors. They're not static, they're dynamic and dynamic states, and being able to observe those would be quite informative in terms of an intelligent design for drugs, and some other things that are listed here. So I think what I've tried to do is pretty obvious, as I indicated before, to show how precision and innovation are important in making new discoveries about nature, the laws of nature, turn that discovery into health, and beginning or I should say continue to push the frontiers of discovery as we began to realize this promising era of precision medicine. I'll leave you with this quote that I'm particularly fond of, in that it is not only insightful, but it also comes from a molecular biologist who is talking about the value of technological innovation. As much as new ideas, Shirley Tillman stated in one of her addresses, are fundamental to the advancement of science, technological innovation is the engine of scientific progress. I thank you for your attention.
So I have no doubt it was going to be inspirational. Really, I think this is really a testament of two things. Number one, Rod's vision, and what also is the NIH and NIBIB with an investigator is pushing. It's very challenging. The bar you put in there, 100 billion frames a second. I see Dan sweating. Matthias, it's like, why did I even hear this? But this is exactly what we wanted to hear, exactly, I think, where the inspiration by the young people and the next innovation should come from. So really, I wanted to thank you. Before opening to question, we also want to give you a little testament to the many, many awards that you get. Hopefully you have a place for us for this. This is to thank you for coming, Rod. Thank you so much. So why don't we have some questions from people? I know you're all in awe, but please, if you can come up to the podium. Maybe I get started. Scott, I think you want to say something? Or no? So maybe I get started a little bit. So as you, since you are privy, and NIBIB is investing a lot, they have a large portfolio of research innovation. Can you maybe give us one to maybe three things from what you think imaging-wise and impact to medicine and translation? Where do you see the areas? Without putting you on the spot, I know you have a huge portfolio. You can't favor one versus the other, but just give us a sense. Where three top things that you believe people here should be thinking about?
Well, I tried to touch on some of those in my presentation. There's no dearth of big problems to address. The large problems that we face thematically are in the area of being more value-driven in our approach to medicine. I think that is a sort of a fundamental challenge we have. The issue of increasing access to modern approaches to diagnostics, modern approaches to therapy is a big one, so that people, independent of where they live, independent of geography, independent of socioeconomic status, they still get these modern diagnostic and therapeutic approaches. And then the broader challenge that we have as a community is to do that in a cost-effective manner, which we all are aware of with the Affordable Care Act, actually underscores that and is trying to drive the health care system towards delivering value. Where this value index I would define as increased quality, decreased cost, and decreased complexity. So the more that we can reduce the complexity of approaches, I think the better. Now, having said that, MR is a technology that is very rich in its variability, rich in the various things that it can do. And no doubt there are many instances where you would like high sophistication. And when those problems occur and they're identified appropriately and can only be solved with that sort of a sophistication. We need to be ready to deliver that. So I think moving towards a lower complexity doesn't mean that we still don't have very sophisticated systems. We will need them both, but we need to be wiser about how they're used and when they're used, and the sophisticated approaches are used when they're needed, and perhaps not for the simpler things. So I think there is a role for both, but I think the broader challenge for this community is to meet these competing demands of making modern technology available to a broader, well, not just broader, to everybody, everywhere.
I mean, you touched in here on a very important point. I think you mentioned two things that resonate, for us is the aspect of value and healthcare. The other aspect, which I think you imply a little bit, is the aspect of frugal innovation. There's a huge push in trying to figure out, we in general, have solutions. We look at, expensive equipment and technology, but at the same time, I think we should think about how can we innovate, cheaper. And that probably is nice for NIH, because this way they can support more people with the money that is available. Dan, please.
So, Rod, first of all, thanks again for a characteristically inspirational presentation, giving us lots of food for thought, and not just in the billion frames per second. But I thought I was detecting a little bit of a theme emerging in some of the examples you used, particularly the ones focused on imaging, which was the importance and the growing prevalence now of mapping, not just in space, but of in space and time. I saw a number of things labeled as four-dimensional, and certainly you can extend to 5 and 6 and so on. I wonder if you have any thoughts about that as a theme, that as maybe even potentially a kind of call to arms for those in imaging to try to develop technologies that now can map many things simultaneously, map large areas, but with fine temporal resolution, and then perhaps the consequences when it comes to big data that will emerge from those kinds of approaches.
Okay, a rich question. Well, indeed. In fact, I've advocated for this four-dimensional thinking, sort of broadly in NIH, and we have a common fund initiative at the NIH, which is called the 4D Nucleon. It actually began as the 3D nucleome, and I said, actually, it should be 4D because it's not static. As I mentioned, nuclear structures don't just sit there. They change in location, they change in conformation over time, and we need to understand that. So the 4D nucleome underscores that and recognizes it. The other thing that you talk about is sort of having this huge and very rich data set from which you could extract a plethora of indices and kinds of information. And that, in part, is the theme of big data across the whole federal landscape. The NIH perhaps leading the way, but this is broadly an approach and taking advantage of the wealth of data that we are acquiring and recognizing that we have more data than we understand, don't always know what's in there, but having this allows us the opportunity to make those kinds of discoveries. The potential to gain new knowledge through that, and also the potential to have a more efficient approach if you can capture a lot of data as all at once, as opposed to sequential prescriptions for individual subsets of data. You capture all this data at once, and then after the fact, go in and pull out what you need with additional follow-up components as needed. That's certainly true in imaging, but it's also true in other things, sort of broadly speaking, I think are true with genetic and genetic relationships. There's so much there that isn't known, a lot more that's unknown, I think, than is known, and the rash of discoveries, the rate of discoveries that keep occurring, I think, are a testament to that.
Matthias,
Rod, thank you very much. Very inspirational. It seems to me that engineering is really at the base of many of these innovations. So it's a crucial topic. Yet, as we all know, there is no Nobel Prize for engineering. So as engineers, we are always a little bit, the lost son. They are not taken that seriously. And in response to that, I think the Whitaker Foundation had a great initiative many, many years ago, and I think many in this room were beneficiaries actually of this Whitaker Foundation money that took engineering seriously and funded engineers. So what's the future of that? Are we going to a more biologically driven society where innovation is done in biology? Is engineering still something that is important and should be valued with such initiatives as the Whitaker Foundation, or even from an NIH point of view?
So a couple of things in that question, Matthias. The importance of engineering, and then the Nobel Prize, and the lack of a Nobel Prize. So I think, the current perspective is changing. Engineering is increasingly recognized by non-engineers as being important. The perspective piece that I referenced in Science Translational Medicine was written by 4 engineers, but it reflects the trend across the nation. An example of that, I can give you a lot of anecdotes from discussions I've had with Harold Varmus, who recently left the NIH as NCI director, about the importance of bringing in physicists into cancer and cancer biology and discoveries, and the importance of what he calls the outer reaches of engineering, where you try and change the behavior of immune cells so that they are more active in identifying and targeting cancer cells, which can be done and is beginning to be done in laboratories, as an example. But a really concrete example of this is that just this year, the University of Illinois has announced the creation of a new medical school targeted in 2017 that is based on engineering, explicitly so. They have a strong engineering school. That's what Paul Lauterbur was, as you know. And this is a movement whose time has come. I've spoken with multiple other universities that have medical schools in engineering about finding a greater intersection between the two and merging. So I think the value of engineering in medicine is being appreciated and is growing, and I also underscored that by the growth in funding from the NIH that's going to engineering departments. Now, the other thing you bring up was the lack of a Nobel Prize for engineering. And I think that's more historical than modern day. I don't know if the Nobel Committee has the wherewithal to add an additional prize. I don't know how that's constructed. I see a head shake no. It's probably based in some statute or so forth, or maybe they could revisit it. But the National Academy of Engineering established through the Russ family a prize, the Russ Prize, specifically because there was no Nobel Prize, and it's equivalent in funding. It is very rigorously reviewed and awarded just every 2 years. It's about a $500,000 prize or so, specifically for high-impact engineering achievements to make a difference in people's lives. The winner of that prize this year, for example, was the team that developed the cochlear implant. So I think those are my responses. It would be nice if there was a Nobel Prize, but I don't know if that's possible. And then there are other, big prizes that cite engineering. The Lemelson MIT Prize, as I mentioned, is one. And then there's a Millennium Prize that cites technological innovation, which Bob Langer won sort of this year. So there are Nobel equivalent prizes in terms of funds, but the Nobel is still, when you're the first, you're always the first. Perhaps you could write them, Matthias.
We'll take a quick question, and then we'll conclude that.
Sure. So you introduced us to a lot of technologies and research that are improving precision. Where would you say technologies and research that are improving accuracy fit into that kind of spectrum of innovation?
Well, I thought there might be a tried and true scientist in the audience that would draw a distinction between precision and accuracy. In the public arena, I think, certainly when the president talks about this, precision and accuracy basically are interchangeable. He's really talking about being accurate in identifying truth. And I think that is the goal. Parsing the differences, the technical differences between precision, which has to do more with repeatability, and accuracy, which has to do with truth, really don't come into play in the public discussion.
You can tell our engineers in the audience.
Hi,
Final question quickly. Yeah.
hi, thank you. I guess this is not working, but anyways, thank you for that wonderful talk. I sympathize with the Nobel Prize engineering issue. I trained as a chemical engineer back in the day. And, but there is some hope. As you may recall, 1979 Nobel Prize in Physiology or Medicine, Cormack and Hounsfield, CT scanning. So, I think there is, right? Let's hear it.
There's also an MRI
Nobel
Nobel Prize.
Okay.
Well, just to comment on that, I actually had that in my train of thought, but I was going on so long. But you're right. I mean, there have been technology-based prizes that have occurred. That was certainly one of them. MRI was another. And then also the super-resolution microscopy. That's, engineering. If you ever hear Eric Betsick give a talk, it's like a classical engineer's talk. He really talks about trading off competing challenges in order to achieve what they were trying to achieve. So how could we get smaller and smaller resolution? Well, you take half of this, and then you give up a little of that, and and so forth. And we couldn't see it from one direction, so we did two things and made from opposite directions. I mean, his talk is very systematic like that, sort of a step by step. Here's what we did in order to achieve an improvement of a factor here, and so forth. So I think it's occurring, although it's not called an engineering prize.
Okay. Thank you so much again.